Today I want to share with you some of our work in studying the T-cell responses in acute and persistent COVID-19. I'm really in the context of cancer patients, because a lot of these patients are on immunotherapies, and which gives us a unique lens to understanding the immune response to infections. First, as human science, especially in COVID-19, requires so much collaboration between basic and clinical groups. I want to acknowledge all the ones who contributed, and we received tremendous help. And I think this is what makes our work so exciting. And the work from my lab was really driven by two stellar postbac students, Nick Han and Justin Kim, who is currently in medical school. And we have very close and ongoing collaborations with Memorial Sloan Kettering, with the laboratory of Santosh Bardana and Ben Greenbaum, and their trainees, David Balls and Olga Udovik. But especially the first part of the talk, which is during the first wave of the pandemic, the infrastructure at Penn was so critical, and we so much helped from people like John Weary, Nuala Meyer, and Scott Hensley in terms of like an institutional support, in terms of standardization for sample processing, recruitment of COVID patients, and assay development, and a lot of support from Huang, and you'll hear about some work from Aaron Bange. So my focus, my interest actually is in the effective checkpoint blockade on T cells, and especially the PD-1 pathway. These are inhibitory receptors you think about as brakes on T cells. And so a lot of our cancer patients are on checkpoint blockade. And what happens is that if you give them a dose of PD-1 blockade, within 7 days, you see a burst of immune response. And you can measure this by very simple cell cycle markers such as Ki-67. And then it kind of plateaus over time. And then the result of this is a 3 to 5-fold increase in your CD8 T-cells in the tumor within 3 weeks. And then for many of these patients, complete responses, or even remission, and potentially cure. And so this is where I was interested in studying for my lab, but I started my lab in the middle of the pandemic. And so really you can't, with the lab shut down, you can't really do any research except for in COVID. And so a lot of our labs, including my lab, shifted to a focus on studying the immune system in COVID, which is extremely informative in terms of cancer as well, because what we learned in cancer is actually derived from immunology, from inflammation, from virology and microbiology and basic immunology. So work from Penn and others have really shown a distinct immune signature in patients with severe COVID-19. And if you take a number of immune parameters, dozens to hundreds, and then you can compress them into a two-dimensional map here. And we can see is that the patients with severe COVID-19 have a very distinct immune signature or fingerprint compared to patients who have recovered or have not been exposed to COVID. And if you take a look at what type of cells contribute to this fingerprint, and you can see things like elevated innate immunity, like neutrophils, robust plasmablast response. Many of these patients have robust antibody responses and highly activated adaptive immunity, including CD4 and CD8 T-cells, but at the same time, profound lymphopenia, showing a depletion and perhaps death of these activated T-cells. And the further work has now kind of elucidated what is the underlying immune mechanism of severe COVID-19. And this is really well set up by talks from Dina and Alba, really showing that your whole immune system has to be intact. Right? There is a reason why. And ordinarily in acute infection, you have this burst of innate immunity that limits the virus and essentially allows your adaptive immunity in your T cells and B cells to kind of clean it up. But in severe COVID-19, there is a defect in the innate immunity, which allows the viral load to go uncontrolled. And therefore, your adaptive immunity is now a step behind. And now, because of the persistent inflammation and antigen load, your innate immunity keeps on increasing, turning into myeloid cells and inflammatory mediators and so on, resulting in immunopathology. And this is particularly relevant in cancer patients. And cancer patients have extremely high mortality. 1 in 4 patients die from COVID-19. And so because of this, we were really, really concerned. In particular, we were concerned about our checkpoint blockade patients. Because as you might remember, the job of the checkpoint blockade is to enhance immunity, right? And especially adaptive immunity. And what will happen to these patients if they get COVID-19? And so we actually designed a prospective study called COVID-19 Outcomes in Patients with Cancer, or COPE. And so it was a prospective study, multi-site across the Penn health system to track these patients and to understand their outcomes. And it was pretty simple. These patients have diagnosed with cancer, they were PCR positive and are hospitalized in the UPHS system. And we were able to enroll 100 patients that was admitted in the UPHS system. Biospecimens were collected, and as you'll see here, we resulted in a strong collaboration and validation cohorts with Santosh Vardhana. The long story short, thankfully, it appears that PD-1 blockade really is not associated with increased severity of disease. In fact, we had very few patients who were admitted on checkpoint blockade, and that was a great thing. But the one thing that we did learn from the study is that hematologic cancer patients clearly had a poorer outcome. And when we looked at, these are clinical labs that we routinely draw, they have elevated inflammatory markers like ferritin, IL-6, and LDH, which made us think that this increased mortality may be due to a dysregulated immune system in these patients. And so we wanted to study what are the immune parameters that determine whether hematologic cancer patient ultimately recovered or succumbed to the disease. And so we leveraged a study led by Nuala Meyer. It's an institutional study where, like, every patient that was admitted with COVID-19 at Hospital University of Pennsylvania was enrolled if they consented, and then had serial biospecimens collected. And we looked in particular at the 22 patients with active cancer compared to the 108 patients without cancer. And as expected, the cancer cohort was a little bit older. They had a female predominance. Most of our cohort was actually Black, and the mortality rate in cancer patients was 3-fold higher. And these samples were studied using 26-parameter high-dimensional flow cytometry paired with serologic analysis. And very early on, what we noted was that these hematologic cancer patients were not making antibodies to SARS CoV-2. And so this led to two questions. One, why aren't they making antibodies? And two is, what is the impact of one arm of the immune system being down, especially as we know that all these units of the immune system is important. And so we use our flow cytometry data to really analyze, to study this question. And so our panel had, of course, your basic immune marker, but the majority of it were markers defined to understand T-cell transcriptional states and B-cell states. Inhibitory receptors, co-stimulation markers, memory effector markers, and so on. But despite that, when you compress all that information into a two-dimensional immune map, the cells really clustered into their distinct cell lineages: CD8 T-cells, CD4 T-cells, and B-cells, what we focused on. And moreover, when you do this, for every single patient, you now have an immune map of each patient. And so to do this analysis, all the panels have to be completely exactly the same. Everything has to be QC'd correctly. So we used 45 of these patients from the cohort to do this analysis. And then also, because each of these patients have an immune map, now we can use what we call an Earth mover distance algorithm, which is an algorithm designed to calculate the how much energy is needed to normalize two maps. And so you can imagine two very different immune maps will have a very high EMD score because it requires a lot of energy to normalize them. And so when you do this for every pair of patients, these 45 patients, you can see that the similar patients will now have similar EMD scores, and it comes into clusters or groups of patients with distinct immunophenotypes. And so what we found is that with 5 different clusters of patients, and they were largely driven simply by the balance of CD4, CD8, and B-cells. And so here, cluster 1 on one hand, you can see there's a depletion of CD4 and B-cells, and you have preserved CD8 T-cells. And on the other side, cluster 5, you have plenty of B-cells, but no T-cells. And this is highlights one of the utilities of we call it unsupervised analysis, where an algorithm does some of the analysis for you, because it helps you not miss the things that are most simple. Like we had expected that it would be a differentiation states, different types of B cells or T cells, and that would drive the differences between patients. But altogether, it was comes down to a balance between a ratio of these different major cell types. And this becomes important because when we look at this EMD cluster 5, who have plenty of B cells but no T cells, we see that these patients are able to make SARS CoV-2 specific antibodies, but their mortality is very, very high. This highlights the point that humoral immunity alone is not able to control acute infection, and that T cells are important as well. From a different direction, we got hints from anecdotes for patients from hematologic cancer. It was very difficult to get samples from these patients because many of them died. But for a few, we were able to get, they were able to recover. We were able to get biospecimens while they were hospitalized. And for these patients, we realized is that they really had no B cells and very few CD4 T cells, but they had intact CD8 T cells. And when we looked at this, when you look at activation markers such as HLA-DR and CD38, and this led to the hypothesis that are CD8 T cells compensating for the impaired humoral immunity in hematologic cancer patients. And so we want to test this hypothesis in a larger group of patients. And so we started a collaboration with Santosh Vardhana at Memorial Sloan Kettering Cancer Center, and they had a cohort of patients with a large number of 45 patients. And as you can see here, the mortality rate was much higher as well in the hematologic cancer patients than the solid. And we utilized their 12-parameter clinical grade flow cytometry. And what they're able to do is just simply order a basic flow cytometry panel, and which was very consistent with serologic analysis. And because of time, I'll just summarize some of the key findings of this cohort is that if you do the same EMD analysis and cluster grouping, you see the same T-cell depleted phenotype that was highly associated with mortality. But we wanted to look specifically at a cohort of patients that were have received anti-CD20 therapy. So they had depletion of their B cells. And indeed, anti-CD20 therapy was associated with depleted B cells and antibodies. And then in this cohort, the anti-CD20 was not associated necessarily with increased mortality. Of course, there was a large spectrum of clinical heterogeneity. But this cohort allowed us to directly test what is the result of the T-cell responses when humoral immunity is absent. And what we saw was that the patients who survived the infection had a higher number of CD8 T cells compared to those who did not. And there was really no difference in the CD4 T cells. And of course, this is in a relatively small number, but we given our preliminary data in the hematologic cancer patients, and we thought that this would extend to the vast majority of hematologic cancer patients, and that overall, heme cancer patients will have a defective humoral immunity overall, and mainly because of 2 points. Is one, a lot of the disease in hematologic cancer patients is in B cells, for example, in B-cell lymphomas or multiple myeloma, so they would be qualitatively, if not quantitatively, deficient. But also, a lot of the patients are on high-dose chemotherapies, and that would not be great for the B cells either. And indeed, when we extend this to all hematologic cancer patients, this trend holds with higher numbers of CD8 T cells in those who survived compared to those who did not. No change in CD4 T cells. And instead, actually, the patients who did poorly had robust B cell responses. And then by using a statistical cutoff, you can identify a cut point where patients above a certain threshold of CD8 T cells did far better than those who fell below that threshold, possibly providing a way to stratify patients in terms of their recovery. And then finally, we want to kind of assess if this is due to antigen-specific responses. And many of the hematologic cancer patients had interferon gamma or IL-2 responses to SARS CoV-2 peptides. But really, what's striking is that the patients who received anti-CD20, all 8 out of 8 of them had antigen-specific responses, antigen-specific T-cell responses, showing that there a lot of, at least a significant part of this immunity is driven by antigen-specific T-cell immunity. And so for this part of the talk, we found is that hematologic cancer patients have defective humoral immunity, and that CD8 T cells are more activated, and those they compensate for defective antibody responses. And a greater number of CD8 T cells are associated with survival. And so we went on to go ahead to understand what happens to these hematologic cancer patients that survive severe COVID-19. I mean, they're able to survive, but we didn't think that having no B cells or antibody responses, and we're sure that they had immunologic and perhaps clinical consequences. And so this part of the study, we have continued in very close collaboration with Santosh and Ben at Memorial, and it was led by Olga, Justin, and David as part of a multi-lab collaboration. And we studied a cohort of patients at Memorial Sloan Kettering Cancer Center, and we called a convalescent cohort. And during the time where otherwise, if you're going to recover, you have probably recovered. And so we defined this as after 30 days of diagnosis. And this cohort had 100 patients. Half of them are hematologic cancer patients, and many of them were very sick. Over close to 2/3 of them were hospitalized, especially in the hematologic cancer patients. A quarter of the hematologic cancer patients were in the ICU. And of note, a third of these patients had prolonged viremia beyond 30 days of diagnosis. A lot of these patients were B-cell lymphoma patients, and many of them received anti-CD20, 41%. And so one of the as we had predicted, patients with hematologic cancers had a blunted antibody response. We look at nucleocapsid antibody responses for solid cancer patients, which reports have emerged that in a big picture way, they have a relatively intact immune system. And you can see the antibody responses peak up to 100 days and then kind of wane. But hematologic cancer patients, even without anti-CD20, have a blunted antibody response, and anti-CD20, you really lose this antibody response. One of the consequences of this, and this has been published as well from the Wardana lab, is that anti-CD20 is associated with a persistent viremia. And so for solid cancer patients, by 30 days, for the most part, they do fine, they go home, and we actually don't check their viral loads. There is some selection bias. But then patients with anti-CD20 therapy, a lot of times they're still in the hospital. They still have a virus, even up to sometimes 100 to 300 days. And so we're assessing the viral load. We can quantitate it using what we call a cycle threshold, where the number of PCR cycles that are needed to detect the virus. And so if you need a high number of cycles, that means you have a low amount of virus, and vice versa. And it being quantitated here, and indeed hematologic cancer patients have a longer duration of PCR positivity, and that we think this is linked to the humoral immunity, because when you look at the cycle threshold value, it's correlated with the amount of antibody responses. So for those who make robust antibody responses, their cycle threshold is high, which means they have a lower viral load. And so then our question is, what happens to the cellular response upon persistent viremia? And so we took their PBMCs, their blood samples at the convalescent phase, and looked at their cellular immunity with similar ways, and using flow cytometry and high dimensional analysis. And we can see the similar clusters. One of them, like cluster 1, that is CD8 T-cell biased. And this is important because most of our patients with prolonged viremia fall in this EMD group 1, and which had a higher number or higher frequency of CD8 T-cells. And then you can quantitate this, and indeed, our patients with prolonged viremia had a higher percentage of CD8 T-cells. And so we want to understand what's happening to these CD8 T-cells as you enter from perhaps what you call acute phase to maybe to a more chronic phase. And do these CD8 T-cells become more dysfunctional, perhaps even exhausted? And so we take the CD8 T-cells and we isolate them and do high dimensional analysis and clustering analysis as well. And we use the algorithm to define what cells are similar to each other. And on the left side, what summarizes that are the more memory-like clusters. On the right side are more effector-like, or cells that have more capacity to have a cytokine function and perhaps to kill. And then we see here is a patient with prolonged disease. They actually have a shift to the right side of the map, and showing an enrichment of effector-like CD8 T-cells. And if you quantitate this, indeed, there's a depletion of memory-like cells and effector-like cells, and an enrichment of effector-like cells with prolonged viremia. Showing so far, persistent disease results in a greater number of CD8 T-cells, and at least phenotypically, what we expect to be highly functional CD8 T-cells. And we really didn't find any significant difference in the CD4 T-cells at this stage, although there was a lot of heterogeneity. And then we also wanted to understand the kind of the antigen-specific responses. And so we did ELISpot peptide stimulation with SARS CoV-2 peptides. And we see that in the patient with prolonged disease, we do have robust interferon gamma responses, and especially in the patients with anti-CD20. And showing that the phenotype that we see in acute really persists even into the convalescent phase, that these CD8 T-cells are still active. What we surmise is that they seem to be responsive to the virus. And so at this point, in our unpublished data, we found that essentially these CD8 T-cell responses continue to seem to be robust. And this ends with an open question still is, and I guess, so why can't patients clear SARS CoV-2 despite robust CD8 T-cell responses? And Is there something with the CD4 T cells that we haven't identified? Or is it simply that and that, of course, B cells and antibodies become very, very important. And if you can't stop the virus from replicating, right? It's like almost a never-ending cycle of invasion. And so we're trying to clarify that using some TCR sequencing data, using like there's an adaptive bioscience database that's annotated for T cells responsive to class 1 and class 2 SARS CoV-2 peptides. And so these are our work that's ongoing. And so with that, I will take any questions.
All right, Alexander, thank you so much for that. Very cool. So let me start and ask. So you had this very, very diverse responses where you have these 5 clusters that are so distinct from each other. One, high in B cells, one high in T. What do we understand anything about the basis of that? Of those clusters? What are they? Is that just patient variability, or is there something we can understand about what causes those?
No, and I think that's what's really fascinating about it. So we know, I mean, some of it is, as you expect, like, I didn't touch upon this, but the advantage of our initial cohort was that it had cancer and non-cancer patients. And a lot of the non-cancer patients were in, we call more balanced immunity, and with not necessarily too high or too low, but sufficient and balanced CD4, CD8, and B-cell responses. And those are the patients who had the lowest mortality. And then if you go into the memorial cohort, which is an exclusively cancer patient, what's interesting is that that normal population is missing. So there's only 4 clusters in memorial. It was actually pretty neat to see that. And so suggesting that there is still some defect, even in the solid cancer patients, although probably more subtle than the hematologic cancer patients. But some of the data that I haven't had time to show is that these distinct phenotypes have very, very strong immunologic signatures. Like, for example, the CD8 biased clusters have very clonal CD8 T-cells by TCR sequencing, and they have some difference in the breadth of immune response, and the CD4 T-cells as well. And that is more clear than just persistent and non-persistent disease. And so there's something more fundamental than even having persistent viruses driving these phenotypes. And yeah, we haven't really got to the bottom of it yet.
So the other question I had was, of course, the anti-CD20 response is easy to understand. You have no B cells left. You're not going to have antibodies, and all the consequences of that are clear. But I wondered, do you see similar things with patients that had more conventional treatment, like rituximab or something? I mean, do those patients also have the same issues as those treated with anti-CD20, or?
Yeah. And so they're broad across the class. And so when we do anti-CD20, they include rituximab and any of other classes. We've even looked into some like BTK inhibitors and targeted therapies and so on. But I think what's interesting is that there is a difference between that and the more aggressive lymphomas that require chemotherapy and anti-CD20. Right? And so the most robust T-cell responses, as you expect, would be in the anti-CD20 therapy alone. And once you go into the more aggressive disease that require chemotherapy, also the myeloid cells, these CD8 T-cell responses are blunted. And so cytotoxic chemotherapy is not good for the T-cell response as expected.
So then you really could lose everything and have no response. Yeah, that would be bad. Yeah. I had one other question, which was, I was thinking PD-1 treatment, I think of for a particular purpose, and I could see it being bad in terms of triggering inflammation and so forth. But I wondered, is it ever used as an antiviral? I mean, do people ever consider stimulating immune responses with PD-1, for viremia?
Yeah, and so no, I know that they've actually done it, I think, in HIV, and I'm actually blanking on the results of that study, but the answer is yes, they have, and I don't think there was any, at least, it was, I think, relatively safe, at least. I don't remember the viral load data.
Yeah. Anyway, interesting. All right. Let's see. Are we having others?
Steve?
Yes.
Dina has a question.
Okay. Yes.
Yeah.
Yes. Go ahead, Dina. Are you? Yeah, you're good to go.
Thank you, Alex. So I've just got a couple of questions on the same sort of theme. Did you concentrate on this sort of CD4, CD8 B cells because there was no differences in your innate cells, NKs, for example, or is it just because that was what your panel was? And in the same vein, did you look at gamma deltas T cells?
No, you're right. The reason why is my focus and expertise was on the T cells, and there's probably profound changes in the innate immunity, and I think our colleagues at Penn, Michael Betts, have published a little bit about it, and there was changes in, I think, CD16 expression on myeloid cells, and perhaps some changes in NK cells. So there's definitely some innate changes that I didn't focus on. We have not looked at gamma delta T cells.
Okay, thank you.
Yep.
Okay, we have one more from Rory DeVries, and I'll read it to you if you want. So it's in your last LA spot, looking for SARS CoV-2 specific T cells, you directly compared non-anti-CD20 with patients on anti-CD20 therapy. And the question is, would the relative amount of T cells in 1 million PBMC from a patient on anti-CD20 therapy explain the difference that you see in virus-specific responses?
Yeah, no, that's a great point. So we've done all this analysis. We normalized to like T cell frequencies and so on, and they're consistent.
Yeah, so I guess the question is just, are you normalizing the fact that there's different T cells in those two populations, right? And the answer is yes, you are. So
yeah, and in fact, the patients who are not on anti-CD20, who are especially patients, a lot of them are on chemotherapy. They are a lot of times more leukopenic. So you actually have an increased number of antigen-specific T cells in the anti-CD20 arm.